Hi everyone, welcome to my presentation from Spider-Man to Superbugs, the magic and reality of bacterial transformation. When I was a kid, I really loved Spider-Man, and there was a clip in the movie that really attracted me.
Sweetie, you're just in time for dinner.
Hey, how you doing, buddy? How was the field trip?
I don't feel well. I'm gonna go to sleep.
You won't have a bite?
No thanks. Had a bite.
Well, did you get some pictures, Peter?
I got a crash. Everything's fine.
What's that all about?
As you can see in this clip, Spider-Man's DNA was rewritten because of the radioactive spider's bite. It's an exciting idea. Can we change DNA to gain new powers? The answer is yes, but not in the way you might think. In our Bio-Wanton lab, we gave superpowers to bacteria through a process called bacterial transformation. Before we get to the experiment, let's understand four key concepts. The first, plasmids. These are tiny cellular DNA molecules that carry special genes. Also, they replicate independently. The second is competent cells. Bacteria treated to be ready and willing to take up foreign DNA. The third is heat shock, a brief 42-degree treatment that helps plasmid enter the cells. And the last one, selectable markers, genes that let us know transformation worked, like antibiotic resistance. Our mission of this lab is to transform harmless E. coli bacteria with a plasmid called PUC19. This plasmid has two key genes. One is ampicillin resistance. It lets bacteria survive on antibiotic plates. The second is the LAC-Z gene. It produces an enzyme that turns colonies blue on special plates. We set up two groups, one with the plasmid and one without as a control. This simple comparison will let us know if our transformation worked. And here is our procedure of this experiment. Step 1, we mix competent cells with plasmid DNA, and we put them on ice for 20 minutes. Step 2 is a critical moment. Heat shock at 42 degrees for exactly 45 seconds. Step 3, recover in nutrient broth at 37 degrees. This lets the bacteria express their new genes. Step 4, plating. We use 4 plates, with and without antibiotic for both groups. And 5, we need to wait for 24 hours. Science requires patience. So what do we expect from this lab? Based on the theory, here's what we expect to be seen. On plain LB plates, both groups should grow. No selection means all bacteria leave. On LB and ampicillin plates, only the plasmid group should grow, and they should be blue. The negative plasmid group on ampicillin should show no growth. That's our negative control. If we see this pattern, our transformation was successful. And here is my actual results. Just as predicted, both LB plates show a lot of white bacterial growth. The LB and ampicillin plate with no plasmid is completely clear, perfect negative control. And the LB ampicillin plate with plasmid has distinct, separate blue colonies. This is exactly what success looks like. The plasmid entered, so the genes worked. In science, controls tell us if our experiment is valid. So, controls matter. In this experiment, the positive control, growth on LB plates, confirms our bacteria were alive and healthy. It tests our cell viability, and it should show growth. And if there's no growth, that means our entire experiment should be questionable. The negative control, no growth on ampicillin plates without plasmid, confirms the antibiotic worked. If bacteria grew here, it could mean contamination or failed antibiotics. Based on our results, both controls passed. Our results are trustworthy. This experiment isn't just a classroom exercise. Bacterial transformation saves lives every day. Take insulin as example. For decades, it came from animals, but today, we use bacteria to make insulin to save many patients' lives. Let's see this from a video.
Human insulin was the first golden molecule of the biotech industry and the direct result of recombinant DNA technology. Currently, millions of diabetics worldwide use synthetic insulin to regulate their blood sugar levels. Synthetic insulin is made in both bacteria and yeast. In order to get bacteria to make insulin, a number of factors need to be considered. Genes in eukaryotes have introns. Bacteria do not have introns in their genes, and so they do not have biochemical machinery to remove introns. Also, eukaryotic proteins are processed after translation. Insulin is first translated into preproinsulin, which is 108 amino acid long. First 24 amino acids are signal protein, which leads preproinsulin out of cell. It is cleaved off, leaving proinsulin in pancreas for further processing. Protein folds into looped structure as desulfide bridges are made between cysteine amino acids. 33 stretch of amino acid is cleaved off, leaving mature insulin. In order to get bacteria to make insulin, first DNA is made based on protein sequence of 2 insulin chains. A and B. These DNA fragments are the inserted into plasmids. Each DNA fragment is inserted into the beta-galactosidase gene on a plasmid. The plasmids also have tetracycline resistance gene. Plasmids are then transformed into bacteria. Tetracycline is added to kill off any untransformed bacteria. The transformed bacteria are grown, then beta-galactosidase and insulin fusion protein is harvested and purified. The beta-galactosidase part of the protein is cleaved off and discarded. The 2 protein chains are mixed finally into insulin, made from bacteria.
Today, we insert the human insulin gene into a plasmid, transform bacteria, and grow them in giant vats. The bacteria become tiny factories producing pure human insulin. This method is safer, more constant, and can meet global demand. This is from a lab technique to a medical revolution. However, just as the Spider-Man movie says, with great power comes great responsibility. Genetic engineering gives us incredible power. However, how to use it properly should raise our concern. The positive uses are clear. We can use this technology to achieve medical treatments, agriculture improvement, and also environment cleanup. However, we must also consider potential risks, such as unintended consequences, essential concerns, and biosecurity issues. As future scientists and global citizens, we have the duty to use this knowledge wisely, to pursue innovation but with caution, transparency, and a focus on the greater good. That's all for my presentation. Thank you for watching.